Hi everyone, the subject of tonight's video is the cytoskeleton, and these are the fibrous structures you find inside the cell. So pictured on the screen right now are two different cells that have been stained with a special procedure called immunofluorescence, and what they're showing is the nucleus stained in blue, but the stringy fibers that in this first picture are kind of yellow are staining a special fiber inside the cell, part of the cytoskeleton. The picture to the right again has the nucleus stained in blue, and it has two different types of fibers stained. One is green and one is red. So what you want to think about when you're thinking of the cytoplasm of the cell and the inner structure of the cell is it's more like a bowl of pasta than a bowl of soup, because it's formed of all these fibrous skeletal structures. We're going to go through three separate structures that form the cytoskeleton. That's the microfilaments, the intermediate filaments, and the microtubules. And we'll go through the composition, the structure, the function, and the relative sizes of these three different parts of the cytoskeleton. So let's start with the microfilament, which happens to be the smallest one. Microfilaments are composed of actin monomers. Actin is a protein. And so a microfilament is actually a polymer of actin monomers. And that's pictured here. And it kind of looks like this little twisted polymer or rope. The function of microfilaments is contraction. This pictured here is a cell dividing into two cells. And right here where I've drawn the arrow where you see a pinch, that pinch is formed by actin microfilaments that are contracting and squeezing these one cell into two cells. Actin microfilaments also function in movement of cells because they can contract, they can propel a cell to move. These are, in a relative sense, they are the smallest of the three fibers involved in the cytoskeleton. So that's the microfilament is the smallest, involved in contraction, made of actin monomers. The next filament is called an intermediate filament, and I bet you can guess it is the middle one in terms of size. It is intermediate. It is larger than a microfilament. It's smaller than a microtubule. It's composed of various proteins. It's not a polymer. Its structure is more filamentous. You can find one long protein. You can have a dimer of proteins kind of wound together, or even a tetramer formed by two dimers. These filaments function to hold the nucleus and the organelles in place. They maintain cell shape. As pictured here in this diagram, they're very much like what you think of as a skeleton. They're not dynamic. They're not moving around. They are very static structures holding shape and holding organelles into place. And again, they are middle in size in between a microfilament and a microtubule. The largest of our cytoskeleton fibers is called a microtubule. It's composed of protein monomers called tubulin monomers. The polymer formed by tubulin monomers actually forms a hollow tube as pictured here. If you look at the cross section, it just looks like a hollow tube. If you look at it lengthwise, it kind of looks like a straw. Microtubules have a very important function inside our cells. They provide what I think of as the train tracks for organelle movements. Organelles in our cell don't just float around aimlessly. They actually move from place to place or don't move based on either holding on to our intermediate filaments or moving along these microtubules, which function as our train tracks. Microtubules, in fact, are not static structures. They're constantly polymerizing, so forming a polymer out of monomers. So this picture here is showing forming a polymer. And then they can depolymerize or break down back into their monomers. If the cell all of a sudden doesn't need a train track going in one direction, it can depolymerize its microtubules and then form a new microtubule going a new direction by forming a new polymer. So microtubules are very dynamic. They form and unform and reform throughout the cell depending on the needs of the cell and what needs to be moved around. Now, these aren't just the train tracks for organelles to move around. Microtubules also perform the very important function of providing the tracks for chromosomes to move during cell division. In a different form, they are also a component of cilia and flagella. And when I say a different form, they're still composed of tubulin monomers. However, when they form a cilia and flagella, they don't constantly unform and reform. They are more static structures when they are part of cilia and flagella. Relatively, these are the largest portion of the cytoskeleton, and that's your microtubules. So if we looked at all of these fibers, all of these skeletal fibers together in one table, you can see the smallest is the actin microfilaments that function in contraction. The middle ones are the intermediate filaments that are made of various different proteins in the form of a filament. They're very static. They hold the nucleus and organelles in place, and they maintain cell shape. And then you have our largest ones, which are the microtubules, formed of tubulin monomers. These are different than the other two filaments in that they form and reform depending on what the cell needs and the direction it needs to move organelles or other things in the cell, and those are our largest portion of the cytoskeleton. So you should have this information both in your notes and in your organelle organizer, and that's it for tonight.